Well, good afternoon, everyone. Thank you for this kind introduction. I would like, in the next 15 minutes, 20 minutes, tell you a little bit more about the development background of Chondrocelect. First of all, I would like to briefly introduce our company. We are a small biomedical company with headquarters based in Belgium, and our main focus is in regenerative medicine, where we try to look for innovative solutions and local treatments for damaged and osteoarthritic joints, mainly in the knee joint. Our lead product is Chondrocelect, and Chondrocelect is a cell-based product for use in an ACI procedure, and this is the first advanced therapy medicinal product, or the first approved advanced therapy medicinal product in Europe. We also have a second product, which is a medical device, chondromimetic. This will not be the scope of this talk. This is a resorbable implant for the treatment of smaller and osteochondral defects. In our pipeline, we mainly look for complementary regenerative solutions, integrating biomaterials. We are also engaged in stem cell research, and we were founded in 2000 as a spin-off of the universities of Ghent and Leuven in Belgium. With this slide, I would like to illustrate a little bit the focus of the company, where you see at the lower end a time scale indicating the duration of the symptoms, and at the upper end the severity of the symptoms, and where current treatment options, and we've heard a lot this morning as well, mainly focus at the two extremes of these time scales. At one end, an adaptation of the lifestyle of the patient, symptomatic relief of pain through painkillers, or even minimally invasive surgical interventions. And at the other end, the replacement of the knee joint through a prosthesis. Our main focus was to try to find a regenerative solution to bridge the gap between the two extremes by looking for an effective and durable repair of the damaged tissue in order to restore the functionality of the patient's knee joint. Chondrocelect is a cell-based product. It's indicated for the repair of single symptomatic full-thickness cartilage defects of the femoral condyle of the knee. As already mentioned in the introduction, it is implanted through a more classical ACI procedure, consisting of, at the left-hand side, taking of a tissue biopsy from a reservoir-bearing area of the knee, subsequent isolation of the chondrocytes and an expansion through a quality-controlled manufacturing process, and then, in the end, at the right-hand side, the implantation of that cell suspension in the cartilage defect. The goal that we wanted to achieve with the development of this product was to find or to effectively restore functionality of the knee joint, and this by using the patient's own cells. And the way we approached this consisted of a couple of key elements in that development. First of all, we wanted to find a solution to maintain chondrogenic potential of the cells. It's a well-known phenomenon that if chondrocytes are isolated outside the body, put in a monolayer expansion culture, that they will gradually dedifferentiate and tend to lose their memory to form, again, stable cartilage once implanted in vivo. So we wanted to control and really standardize that part of the process. Secondly, we wanted to achieve a robust and standardized manufacturing process that, although we are working with autologous cells, that we control as much as variables as possible, so that in the end we can guarantee a high quality end product. We also wanted to demonstrate clinical evidence of these characterized cells through a randomized controlled trial. And last but not least, we wanted to implement throughout the whole development cycle of the product the GXP principles for the good manufacturing, good clinical, good laboratory principles to ensure the quality, safety, and efficacy. Now, if you take all these key elements together, the step of developing a product as a medicinal product has actually already been taken. So that's why we decided already in 2000 to develop a cell-based product as a medicinal product. Although at that time guidance was not yet really clear, and this would result in a series of challenges that we have to face throughout the development, because it's a bit of a particular product, especially if you compare it to the more classical biological and pharmaceutical products that we know. The end result was a high quality and characterized cell product, which we also put on the market in a controlled regulatory environment. This means there is no gray legal or ethical zone that remains for the application of this product. A couple of illustrations of the development challenges that we faced, and these went through each and every single step of the development process. On one hand, the manufacturing and product characterization, they're the main challenges that we faced was to manage the biological complexity of this type of product. Especially in a world at that time where there was a lack of established markers and methods to characterize that cell population. On the non-clinical development, one of the main challenges, and we heard a lot about animal models already this morning, was indeed the selection of the right animal model to demonstrate the proof of concept and the safety of the product before moving into the clinic. In the clinical development, the setting up of an RCT, there we faced the choice of a competitor. At the time, in 2000, 2002, when the trial enrollment started, there was no other medicinal approach with a similar mode of action, which was available throughout the whole of Europe to do a multinational randomized controlled trial. So therefore, we had to choose another competitor. We took microfracture, as that was at that time certainly the standard of use in most countries. Biological repair process in itself is also quite a long process. So this also means that for such a trial, the follow-up of the patients is another additional challenge, especially if you compare the length that patients are followed up in an RCT for a cell-based product to other classical biological products. And then it didn't stop, and it hasn't stopped at this moment either, because also in the post-authorization setting, once you develop a medicinal product, you have the obligation to follow the current state of the art, to follow the scientific evolution, and to improve the product or the procedures associated with it. In the next series of slides, I will give you a series of examples on how we try to tackle all these different challenges. On one hand, the characterization of the product. To manage the biological complexity of this product, we did an extensive exercise looking at the three main functional biological levels of what we expect these cells to do. First of all, at the tissue level, and this is illustrated also in the picture below, we developed a mouse model where we could inject the cell suspension in an ectopic site and then look whether that cell suspension could still form a stable cartilage tissue, which could then be analyzed histologically. So that was one level. Then we also looked through three-dimensional culture assays at the cellular level to look for the expression of and the generation of marker proteins and proteoglycans that form the essential building blocks of the cartilage matrix. And we also looked at the molecular level to look for the expression of specific marker genes that would encode for these proteins. And by establishing a correlation on these three functional and between these three functional levels, we succeeded to characterize the cell product. With respect to the manufacturing process, we took at the lower end the approach that you would take for every medicinal product, looking at the key quality attributes in each of the different steps of the manufacturing process, actively monitor these, document these, and make sure that these remain within pre-specified ranges. But on the other hand, because this process also requires quite some manual human interventions, we also had to focus very much on standardization to also limit the variability that could be introduced in the manufacturing process by operators, or at the taking of the biopsy by getting bad biopsy material to start with in the first place. As a proof of principle, we chose a goat model in line with the ICRS recommendations. And at the upper end, you see an image of phenotypically stable chondrocyte population, Chondrocelect-like cell population that was implanted in an inflicted cartilage defect in the knee of the goat. We also used a couple of controls, cells that we deliberately let to de-differentiate they could not, in that ectopic mouse model, for instance, make stable cartilage tissue anymore. And also a complete negative control at the lower end fibroblastic cells that have nothing to do with cartilage. And as you can see from these selected images, the Chondrocelect-like cells at 10 weeks showed good filling, quite a good filling of the defect with the start of integration as well, whereas the controls have much worse or even no results at all. And with this information, we then conducted a randomized controlled trial. I will not go into the details because the results of this trial will be the topic of the next presentation. But we compared Chondrocelect versus microfracture, and the hypothesis that was formed the basis of this trial was to demonstrate structural superior tissue repair at the short term in order to have a precursor for long-term clinical benefit. And then we applied for a marketing authorization. In order to do so, there are a couple of conditions that need to be fulfilled. First of all, the demonstrated quality, safety, and efficacy also needs to be documented and needs to be written down in a concise dossier where regulators and scientists can independently make a judgment of the quality of that data. Because that dossier is then submitted to the regulatory authorities, in this case placed or located at the European Medicines Agency, where there is a scientific committee, the Committee of Advanced Therapies, that consists of scientists active in the regenerative medicine field. And this group of scientists come from all the different member states in Europe. They make an independent evaluation of the dossier, and if that is positive, then there is the third step, the legal endorsement by the European Commission. The outcome of the scientific evaluation of our dossier resulted in a positive benefit-risk assessment, but also endorsed that appropriate management of the product in the real-life setting, because of course we have demonstrated that quality and safety and efficacy in the clinical trial setting, and later on the product is now put in the real-life world, to also manage the product in that setting, with an attention for both the product as well as the procedure surrounding it. It's one thing to have a very good quality product, but if it is used or misused in the wrong way, then also the result can be affected, and especially in a complicated surgery as an ACI procedure, also the standardization of that procedure and the subsequent rehabilitation form an initial part of the product follow-up. And of course, we had to make a long-term engagement also to continue to improve that product profile. For the last part of my presentation, I would like to guide you a little bit more through the actual procedural steps and what comes to see with the use of this product, and also go into the training and the technical support that our company has set up. First of all, treatment of the patient starts, of course, with the patient screening, the anamnesis, the indication setting, and informing of the patient of what will happen and what the subsequent steps will be that he or she will undergo. And then in the next step, through an arthroscopic procedure, the biopsy can be taken. For this biopsy, it is imperative that we obtain good quality material to start from. There's a saying that says scrap in, scrap out. This could also be applied to this type of product. If you get diseased tissue material or loose debridement material of bad quality, then of course your starting point, the cells that you need to manufacture your product with, already have a backlog, and this could also end up in a worse quality. The biopsy is typically taken from a lesser weight-bearing area. We have also developed a CE-marked biopsy instrument to try to standardize the biopsy taking as well. This instrument is specifically designed to get a full thickness cartilage biopsy, but without damaging the subchondral bone. And at the same time, a blood sample is taken for virology testing. This is also a typical requirement from the European Cells and Tissues legislation. In case of donation of human body material, virology testing needs to be done. To ensure the logistics, we have developed a standard biopsy kit to make sure that the blood tubes, and also the material, the biopsy material itself, reach our cell expansion facility, our manufacturing facility, in a good shape and in the best possible conditions. Therefore, we also have foreseen a qualified courier service. And this all to ensure that we can work as much as possible with fresh material so that we can do a tissue release within 48 hours of biopsy taking. The implantation then starts with the preparation of the defect bag, the removal of the, and this is through an open knee surgery, removal of the debridement material and diseased material, and the preparation of a physical seal to cover the defect. This used to be a periosteal flap in the past. Nowadays, more and more, we also heard about that. Collagen membranes are used. This collagen membrane is sized to the shape of and the size of the defect, and is then sutured over the defect. And then under that membrane, the Chondrocelect cell suspension can be injected to conclude the implantation step. Chondrocelect is formulated as a suspension in a vial containing 4 million cells. And we deliver, because the dosage is approximately 1 million cells per square centimeter of defect, we deliver a sufficient number of vials, so also a sufficient number of cells to treat the actual defect size of the individual patient. The shelf life of the product, because it's a living product, is very short, 48 hours. But also, therefore, we foresee a standard implantation kit and work with the same controlled and qualified courier service as we did before with the tissue biopsy. Now, you can imagine that with such short turnaround times and different actions that need to be taken, that a good planning is imperative to make sure that unforeseen occurrences would result in non-implantation or a rejection of the product. After the implantation, there is also the rehabilitation of the patient. In our clinical trial, we also developed specific rehabilitation protocols depending on the size and the site of the lesion of the individual patient, so that physiotherapists could easily access or get access to the individual rehabilitation scheme for his or her patient through a central website, and this through a unique patient code that is provided to the physiotherapist. In this way, the physiotherapist has more detailed guidance for the individual particular patient that he can use then to further provide instructions and guide the patient through a good rehabilitation phase. The educational, because of the good planning and the management of the product in daily practice, we have also foreseen an educational program for hospital staff. This foresees on one hand in training on the logistics and the actual handling of the product, but on the other side, on the other hand, also provides information on surgical technique and the rehabilitation associated with it. We've also developed a user manual. This is sort of a guide explaining all the different steps. And we do provide medical information and feedback once the hospital starts implantations with Chondrocelect, so that the surgeon can also further improve his or her skills. With respect to the technical kits for the biopsy harvest and the Chondrocelect implantation, these come with clear user instructions, but they also contain all the necessary specific equipment which is needed, the non-standard equipment which is needed for this procedure. And we already alluded to the uniform and centralized logistics handling. So in overall summary, Chondrocelect is an ACI product. It's a cell product which is developed specifically for an ACI procedure. It's a ready-to-use cell suspension. It's delivered as an individual treatment dose for a specific patient. There is a training and support for practical handling of the product foreseen. And as a company, we have also the commitment to continue to follow up the product, technical improvements in accordance with the evolving science. But also, we follow closely the safety signals that come out of pharmacovigilance follow-up of the patients. And we also committed to further